हॅलो फ्रेंड्स, राणा अकॅडमी या यूट्यूब चॅनलवरती आपल्या सर्वांचं स्वागत आहे. आपण स्टार्ट केली आहे महाराष्ट्र स्टेट बोर्डची बायोची 11th क्लासची सिरीज, ज्यामध्ये आपण आतापर्यंत 4 चॅप्टरपर्यंत पाहिलं. तुम्ही जर या चॅनलवरती नवीन आला असाल तर प्लीज या चॅनलला सबस्क्राईब करा आणि बेल आयकन हिट करा. चला तर मग व्हिडिओला सुरुवात करूया. 5 नंबरचा हमारा चॅप्टर आहे सेल स्ट्रक्चर अँड ऑर्गनायझेशन. लेट स्टार्ट. Cell. Cell is called as a structural and functional unit of life of all living organisms capable of independent existence and can perform all functions of life. To see cells clearly, we need a microscope. Large cells can be seen through a simple microscope, but to see the smaller cells we require a compound microscope. Simple microscope can magnify image 50 to 100 times, but a compound microscope can do so 1000 times or more. In the microscope we use in the laboratory, a beam of light is used to make things visible, hence it is light microscope. To see the interior of cell on need cell we need an electron microscope that can magnify image 5 lakh times. There is no typical shape of a cell. Cells may be spherical, rectangular, flattened, polygonal, oval, triangular, conical, columnar, etc. Cell size varies greatly in various plants and animals. Some of them are not visible to naked eye. Some are barely visible. Size can be seen in mycoplasma 0.3 micrometer in the length. Bacterial cell size is 3 to 5 micrometer, while the largest size of cell is seen in ostrich nearly 15 centimeter cell. Longest cells are nerve cells. You already know that cell theory was proposed by Shawan and Skeldon. However, in this theory, there was no explanation about formation of new cells. It was Rudolf Virchow who explained for the first time that new cells are formed by cell division from pre-existing cells, omnicellula e cellula. And in later years, advanced research in cytology led to modification in cell theory, which is now known as modern cell theory. Totipotency. It is the capacity or potential of living nucleated cell to differentiate and divided to form new any other type of cell, and thereby a complete new cell organization, a new organism. A cell is totipotent because it has a entire genetic information of the organism in its nucleus. Embryonic animal cells are totipotent and termed as stem cells. Stem cells have great medical applications, including cure for diseases. Kinds of cells. Living organisms are grouped into two main categories, the prokaryotes and eukaryotes. The prokaryotes have simple cellular organization, while eukaryotes exhibit high degree of organization. Prokaryotic cells. The cell in prokaryotes show following main features. It has chemically complex, have well-defined nucleus and other membrane-bound cell organelles. A cell envelope is of three layered structure with outer glycocalyx, middle cell wall, and inner plasma membrane. Glycocalyx is present as either slime layer or capsule. Bacteria are better observed when stained. The most followed staining method is gram staining, developed by Danish bacteriologist Hans Christian Gram. The cell wall is made up of peptidoglycan in gram-positive bacteria and murine in gram-negative bacteria. It gives mechanical strength to the cell. Cell membrane is a phospholipid bilayer. All these structures give protection to the cell and also help in intercellular transport. Then motile bacteria Other cilia or flagella are produced by basal body, which works as motor. Other parts are filament and hook. Some other surface projections are the tubular pili, which help in intercellular communication, and fimbriae, or clinging to support. The cell membrane shows in folding called mesosomes, which help in cell wall formation, cellular respiration, and DNA replication. Some bacteria, especially photosynthetic cyanobacteria, show more longer extensions called chromatophores. They carry photosynthetic pigments. The cytoplasm contains dense particles called ribosomes, helping in protein synthesis. Ribosomes are described by their sedimentation rate in Swedberg unit. Bacterial ribosome are 70 s, composed of larger subunit 50 s and smaller subunit 30 s. Figure 5.3, prokaryotic cell. Eukaryotic cell. Cells in which the nucleus has a definite nuclear membrane are known as eukaryotic cells. These cells exhibit presence of membrane-bound cell organelles. Example: cells of protists, plants, animals, and fungi. The eukaryotic cells have different shapes, size, and physiology, but all the cells are typically composed of plasma membrane, cytoplasm and its organelles, viz. Mitochondria, endoplasmic reticulum, ribosomes, Golgi complex, etc., and a true membrane, a true nucleus. Components of eukaryotic cells. Cell wall. It is rigid, supportive, and protective outer covering of plasma membrane of plant cells, fungi, and some protists. Algae show presence of cellulose, galactons, mannans, and minerals like calcium carbonate in cell wall. And other plants, it is made up of microfibrils, hemicelluloses, pectin, lipids, and proteins. Microfibrils of plant cell wall show presence of cellulose, which is responsible for rigidity. Some of the depositions of cell wall are silica, grass stem, cutin, epidermal walls of land plants, suberin, endodermal cells of root, wax, lignin. It gives shape of cell and protects from mechanical injury and infections. Figure 5.4, plant cell. In plant cell wall shows middle lamella, primary wall and secondary wall. Middle lamella. It is thin and lies between two adjacent cells. It is the first structure formed from cell plate during cytokinesis. It is mainly made up of pectin, calcium, and magnesium pectate. Softening of ripe fruit is due to solubilization of pectin. Primary wall. In young plant cell, it is capable of growth. It is laid inside to middle lamella. It is the only wall seen in meristematic tissue, mesophyll, pith, etc. Secondary wall. It is present in the primary wall. Once the growth of primary wall stops, secondary wall is laid at some places. Thickening is absent, which leads to formation of pits. Plasmodesmata are cytoplasmic bridges between neighboring cells, which show the pores between primary cell wall and middle lamella. Figure 5.5, animal cell. Figure 5.6, cell wall, plant cell. Cell membrane, plasma membrane, biomembrane. Cell membrane, plasma membrane, biomembrane. It is thin, quasi-fluid structure present both extracellularly and intracellularly. Extracellularly, it is present around propagules of, and intracellularly, it is present around most of the cell organelles in eukaryotic cell. It is separated cell organelles from cytosol. Thickness of biomembrane is about 7 angstrom under Under electron microscope, cell membrane appears trilamelate, made up of three layers. It is shown presence of lipids, mostly phospholipids, arranged and bilayer lipids possess one hydrophilic polar head and two hydrophobic non-polar head tails. So phospholipids in two layers bilayer in such a way that their tails are sandwiched in between heads. Due to this, tails never come in direct contact with aqueous surrounding. Cell membrane also shows presence of proteins and carbohydrates. Ratio of proteins and lipids varies in different cells. For example, in human beings, RBCs show approximately 52% protein and 40% lipids. Fluid mosaic model, it is most accepted model of cell membrane. It was proposed by Singer and Nicholson in 1972. According to this model, it is made up of phospholipid bilayer and proteins. Proteins are like icebergs in the sea of lipids. Proteins can change their position. Some proteins are intrinsic, that is occur at different depths of bilayer. Figure 5.7 fluid mosaic model. They span the entire thickness of the membrane, so they are called transmembrane protein. They are form channels for passage of water. Extrinsic or peripheral proteins are found on two surfaces of the membrane. Quasi-fluid nature of lipid enables lateral movement of proteins. This ability to move within the membrane is measured as fluidity. Main function of plasma membrane is transport of molecules across it. This membrane is selectively permeable. During passive transport, many molecules move across the membrane without spending energy. Some solutes move by simple diffusion along the concentrations. The neutral solutes may move across the membrane by a process of simple diffusion. This is called the passive transport. Water may also move by osmosis. During active transport, few ions or molecules are transported against concentration gradient from lower to higher concentration. It requires energy, so ATP is utilized as such. Transport is an energy dependent process in which ATP is utilized. It is called active transport. Example: sodium potassium pump. Polar polar molecules cannot pass through non-polar lipid bilayer, so they require carrier of proteins. Cytoplasm. The cell contains ground substance called cytoplasmic matrix or cytosol. This colloidal jelly-like material is not static. It shows swimming movements called cyclosis. Along with organic and inorganic molecules like sugars, amino acids, vitamins, enzymes, nucleotides, minerals, and waste products. It also contains various cell organelles like endoplasmic reticulum, Golgi complex, mitochondria, plastids, nucleus, microbodies. Cytoplasma acts as a source of raw materials as well as seed for various metabolic activities taking place in the cell. It helps in distribution and exchange of materials between various cell organelles. Cell organelles are nothing but compartments in the cell that carry or specific functions. Some of them coordinate with each other and complete specific tasks for the cell. Nuclear membrane, endoplasmic reticulum, Golgi complex, lysosomes, and various types of vesicles and vacuoles form such a group and are together considered as endomembrane system of the cell. The organelles having distinct functions are not included in the endomembrane system. Example: mitochondria or chloroplast carry out specific type of energy conversions in the cell. Endoplasmic reticulum (ER). This little network within the cytosol is present in all eukaryotic cells except the ova and mature red blood corpuscles. Under the electron microscope, it appears like a network of membranous tubules and sacs called cisternae. It forms more than 50% of the total membrane of the eukaryotic cell. This divides the cytoplasma called laminar cytoplasma and non-laminar cytoplasma that ties outside ER. Membrane of endoplasmic reticulum. It is continuous with nuclear membrane envelope at one end and extends till cell membrane. It thus acts as intracellular supporting framework and helps in maintaining position of various cell organelles in the cytoplasm. The outer surface of endoplasmic reticulum membrane or may not be studded with ribosomes. Accordingly, it is called a rough or smooth endoplasmic reticulum. Smooth or rough endoplasmic reticulum differ in their functions. Smooth endoplasmic reticulum is involved in various processes on different cells depending on cell type. It helps in a synthesis of lipids, example steroid secreting cells of cortical regions of adrenal glands, testes, and ovaries. Detoxification of drugs and poisons, liver cells, and storage of calcium ions, muscle cells. Rough endoplasmic reticulum is primarily involved in protein synthesis, example pancreatic cells that secrete insulin. These proteins are secreted by the ribosomes attached to rough endoplasmic reticulum and are called secretory proteins. These proteins get wrapped in membrane that bursts off from the transitional region of ER. Such membrane-bound proteins depart from ER as transport vesicles. Rough endoplasmic reticulum is also involved in formation of membrane for the cell. The ER membrane grows in place by addition of membrane proteins and phospholipids to its own membrane portions of this expanded membrane are transferred to other components of endomembrane system. Golgi complex or Golgi apparatus or Golgi body. Various terms are used to denote this assembly, manufacturing, or packaging, and transport unit of cell. Golgi complex essentially consists of stalks of membranous sacs and called cisternae. Diameter of cisternae varies from 0.5 to 1 micrometer. A cell may have few to several cisternae depending on its function. Now, the thickness and molecular composition of two membranes of a Golgi sac differ from each other. The Golgi sacs show specific orientation in the cell. Each cisternae has a forming of or cis of face cis on the same side and manufacturing or trans opposite side. Transport vesicles that pinch off from the transitional ER emerge from with cis face of Golgi cisternae and add its contents into the lumen. Figure 5.9 Golgi complex. This explains why Golgi bodies are usually located near endoplasmic reticulum. Modified and condensed secretions have leave Golgi through trans phase again as membrane bound vesicles. Golgi body carries out two types of functions: modification of secretions of ER and production of its own secretions. Cisternae contains specific enzymes for specific functions. Refining of products takes place in an orderly manner. For example, glycolipids and glycoproteins that are brought from endoplasmic reticulum lose contain certain sugars and regain others thus forming a variety of products. Golgi bodies also manufacture their own products. Golgi bodies is many plant cells produce non-cellulose polysaccharides like pectin manufactured or modified. All products of Golgi complex leave cisternae from transphase as transport vesicles. While they are living from the Golgi, certain markers may get impregnated or they on their membranes so that they can identify their specific largest target cell or cell or organelle. Lysosomes can be considered as dismantling and restructuring units of a cell. These are membrane-bound vesicles containing hydrolytic enzymes. The enzymes are lysosomes are used by most eukaryotic cells to digest hydrolyze macromolecules. The lysosomal enzymes show optimal activity in acidic pH. Lysosomes arise from Golgi-associated endoplasmic reticulum. The list of lysosomal enzymes includes all types of hydrolysis, while amylases, proteases, and lipases. These enzymes are in inactive state and are activated only when a lysosome comes in contact with another particular organelle to form a hybrid structure. After the action of enzymes is over, the lysosomes is reformed and reused. Lysosomes are thus found in various structural forms and carry out various functions of the cell. Lysosomes are polymorphic in nature. We can classify lysosomes as primary lysosomes, which are nothing but membrane bound or vesicles in which enzymes are in inactive state. Secondary lysosomes are hybrid lysosomes, which are formed by fusion of lysosomes with endocytic vesicles containing materials to be digested. They are represented as hydrophobic vesicles. This is larger in size than primary lysosomes. A residual body in the vesicle containing undigested remains left over in the hydrophobic vesicle after releasing the products of digestion in the cytosol. Lysosomes, which bring about digestion of cells' own organic material like a damaged cell organelle, are called autophobic vesicles. As autotrophic vesicles essentially consist of lysosome fused with membrane-bound old cell organelles or organic molecules to be recycled. Remember, every week human liver cells recycles half of its macromolecules. Lysosomes bring about intracellular and extracellular digestion. The intracellular digestion is brought about by autophobic vesicles or secondary lysosomes, which contain foreign materials brought in by processes like phagocytosis. Example: food waste vacuoles and amoeba and macrophages in human blood that engulf and destroy harmful microbes that enter the body. Extracellular digestion is brought about by release of lysosomal enzymes outside the cell. Example: aerosomes can a cap-like structure in human sperm is a modified lysosome which contain various enzymes like hyaluronidase. These enzymes bring about fertilization by dissolving protective layers of foam during metamorphosis process found in many organisms. Lysosomal enzymes help in reusing the tissues of redundant organs. They also help in destruction of malignant cells. Example: T lymphocytes. Vacuoles are membrane-bound sacs prominently found in plant cells and animal cells. Whenever present, they are few in number and small in size. Generally, there are two or three permanent vacuoles in a plant cell. In some large plant cells, a single large vacuole occupies the central part of the cell, and it is called a central vacuole. In such cells, vacuole can occupy as much as of the 90% of the total volume of the cell. The vacuoles are bound by semi-permeable membrane called a tinoplast membrane. This membrane helps in maintaining the composition of vacuous fluid. The cell sap different from that of the cytosol. Composition of cell sap differs in different types of cells. The cell sap is of central vacuole is a storehouse of various ions and thus the two hypertonic to cytosol. Small vacuoles in seeds of certain plants store organic material like products. Other compounds are stored that are harmful or unpalatable to herbivores, thereby protecting the plants. Attractive colors of the petals are due to storage of such pigments in vacuoles. Intake of food and foreign particles by phagocytosis involves formation of food vacuole in fresh water. Unicellular forms like paramecia, excretion, and osmoregulation takes place by contractile vacuoles. Vacuoles maintain turbidity of the cell. Glycosomes. These membrane-bound organelles contain enzymes that convert fatty acids to sugar. They can be observed in cells of germinating seeds. There where the cells utilize foods, fats as a source of sugar till it starts photosynthesizing on its own. Figure 5.11 Glycosomes. Mitochondria, singular mitochondria. These are important cell organelles involved in aerobic respiration. Mitochondria are absent in prokaryotic cells and red blood corpuscles RBCs. Their shape may be oval or spherical or spiral strip-like. It is a double membrane bound organelle. Outer membrane is permeable to various metabolites due to presence of protein, purines, and Parsons particles. Inner membrane is selectively permeable to few substances. Only both membranes are separated by a space outer chamber. Inner membrane shows several finger-like or plate, and the folds crystal. Inner membrane bears numerous particles, oxysomes, and cytochromes, electron carriers. Inner membrane encloses a cavity, inner chamber containing a fluid matrix. Matrix contains few coils of circular DNA and RNA, 70s type of ribosomes, lipids, and various enzymes of Krebs cycle and other pathways. Figure 5.12 Mitochondria. Oxysomes. The inner membrane of mitochondrion bears numerous particles. Oxysomes F1 to F0 fertilized membrane of elementary particles, mitochondrion particles. Each particle consists of head and stalk, foot, head lollipop, head of faces towards matrix, and foot F naught is embedded in inner membrane. Head acts as an enzyme ATP synthase and foot has proton channels. Oxysomes are involved in proton pumping and ATP synthesis. Plastids like mitochondria. Plastids two are double membrane organelles containing DNA, RNA, and 70s ribosomes, but they are larger in size and can be observed under light microscope. Plastids are classified according to the pigments present in its. It has leucoplast, Now, chromoplasts, chloroplasts, leucoplasts do not contain any pigments. They are of various shapes and sizes. These are meant for storage of nutrients. Example: amyloplasts and that store starch, eleoplasts that stores oils, and alleroplasts that stores proteins. On figure 5.13, chloroplast. Chromoplasts are contain pigments like keratin and xanthophylls, etc. They import red, yellow, and orange color to flowers and fruits. Uh, now you must have understood why potato looks like white in color and shows flower petals are red. Both leucoplasts and amyloplasts do not contain photosynthetic apparatus we find in chloroplasts. Let us now study the chloroplasts in details. The plant cells of algae and few protistas like euglena contain chloroplasts. Chloroplast. You have observed ribbon-shaped chloroplast in Spirogyra. It differs in size, number, and shape of in various cells in which it is found. And plants, it is found in green regions, mainly in mesophyll of leaf. Now, this chloroplast is lens-shaped, but it can also be oval, spherical, discoid of ribbon like a cell may contain a single large chloroplast as in chlamydomonas, or there are 20 to 40 chloroplasts per cell seen in mesophyll cells. The chloroplasts contain green pigments. The chlorophyll, along with other enzymes that help in production of sugar by photosynthesis. The inner membrane of double-walled chlorophyll is comparatively less permeable. Inside the cavity of inner membrane, there is another set of membranous sacs called thylakoids. Now, thylakoids are arranged in the form of stalks called grana, singular granum. The grana are connected to each other by the means of membranes tubules called stroma. Lamellae space outside thylakoids it is filled with stroma. The stroma and the space inside thylakoids contain various enzymes essential for photosynthesis like other plastids. Stroma of chloroplast also contains DNA and ribosomes. Ribosomes. You are aware that ribosomes and proteins factories of the cell. They use the genetic information to synthesize proteins. Ribosomes were first observed as the dense ribosomes were first observed as dense particles in electron micrograph of a cell by scientist Faraday in 1953. The ribosomes are made up of ribosomal RNA and proteins. They do not have any membranous covering around them. Figure 5.14, ribosome. In a eukaryotic cell, ribosomes are present in mitochondria, plastids, and in cytosol. Our ribosomes in cytoplasm are either found attached to outer surface of rough endoplasmic reticulum and a nuclear membrane, or freely suspended in cytoplasm. Both are similar in structure and are ATS types. Each ribosome is made up of two subunits, a large and a small subunit. The bound ribosomes generally produce proteins and that are transported outside the cell after processing in endoplasmic reticulum and Golgi body. Example: bound ribosomes of acinar cells of pancreas produce pancreatic digestive enzymes. Free ribosomes come together to form chains called polyribosomes for protein synthesis. Free ribosomes generally produce enzymatic proteins that are used up in the cytoplasm, like enzymes required for breakdown of sugar. Both types of ribosomes can interchange position and function. Number of ribosomes is high in the cells actively engaged in protein synthesis. Nucleus structure of nucleus of a eukaryotic cell member becomes distinct in a non-dividing cell or during interphase. Such an interface Such an interface nucleus is made up of nuclear envelope, nucleoplasma, nucleolus, and chromatin network. Nuclear envelope is a double-layered the delimiting membrane of nucleus. Two membranes are separated from each other by petty nuclear connected with endoplasmic reticulum and places. It also harbors ribosomes on it. The inner membrane is lined by nuclear lamina, a network of protein fibers and that helps in maintaining the shapes of nucleus. The two membranes along with the perinuclear space now help in separating nucleoplasma from a cytoplasm. However, nuclear membrane is not continuous at places. There are small openings called nucleopores. The nucleopores are guarded by spore complexes which regulate flow of substances from nucleus to cytoplasm and in reverse direction. Figure 5.15 nucleus. The nucleoplasma and carolymph contains various substances like nucleus, nucleic acid, proteins, protein molecules, minerals, and salts. It contains a chromatin network and nucleolus. Nucleolus is another component which is not bound by cell membrane. Nucleolus is made up of rRNA and ribosomal proteins, and it is best known as the site of ribosomes biogenesis. Depending on the synthetic activity of a cell, there are one or more synthetic activity of a cell. There are one or more nucleoli present in the nucleoplasm. For example, cells of the oocyte contain large nucleolus, whereas sperm cells contain small in corpuscles one. They appear as dense spherical bodies present near chromatin network. Now, they are produce rRNA and ribosomal proteins, which are then transported to cytoplasm and are assembled together to form ribosomes. Chromatin material. Chromatin material nucleus contains genetic information in the form of chromosomes, which are nothing but DNA molecules associated with proteins. In non-dividing cells, the chromosomes appear as thread-like network and cannot be identified individually. Now, this network is called chromatin material. The chromatin material contains DNA, histone, and non-histone proteins and RNA. In some regions of chromatin, DNA is more and genetically active called euchromatin. Some regions that contain more of proteins and less DNA and are genetically inert are called heterochromatin. Figure 5.16 ultra structure of nucleus. When the cell prepares to divide, the chromosomes coil and get recognized all at the metaphase stage. Now they are becomes distinct and can be clearly identified. You will study these processes as well as structure and types of chromosomes in other chapters. Every species of living organism has specific number of chromosomes, like normal human cells now has 46. The nucleus contains entire genetic information, hence play important role in important role in heredity and variation. It is the site of synthesis of DNA, RNA, and ribosomes. It plays important role in protein synthesis. The chromosome number being consists of for a species. It is important in phylogenetic studies. Nucleus this is the master cell organelle. Cytoskeleton. When advancement of in light and electrons microscopy, scientists revealed the presence of network of fibrils throughout the cytoplasm. It is called cytoskeleton. Cytoskeleton consists of microtubules, microfilaments, and intermediate filaments. Microtubules are made up of proteins, tubulin. Microfilaments and are composed of fibrous proteins. Cytoskeleton helps in maintenance of shape of cell, contraction of cell, mobility of cell and cell organelles, and changes in shape of the cells and cell division, cilia and flagella. They are fine hair-like membrane bound protoplasmic outgrowths that occur on the free surface of the cell. They generate a current and fluid medium for passage of material and locomotion. Cilia are small in size and many in number. Cilia acts as oars causing the movement of cell. Flagella are longer and few in number. Flagella present in prokaryotes bacteria called are structurally different from that of eukaryotic flagella. Cilium or flagellum consists of basal body, basal plate, and shaft. Basal body is placed in outer part of cytoplasm. It is derived from centriole. It has 9 peripheral triplets of fibrils. Shaft is exposed of part of cilia and flagella. It consists of two parts, sheath and axoneme. Sheath is the covering membrane of cilium and flagellum. Core called axoneme possesses 11 fibrils running parallel to long axis of shows, 9 peripheral or doublets and 2 single central fibrils, 9 plus 2. The central tubules are enclosed by a central sheath. This sheath is connected to one of the tubules of peripheral tubules by a radius spoke. Central tubules are connected to each other by bridges. On the peripheral doublets are connected to each other through linkers or interdoublet bridges. Centrioles and centrosomes. Centrosomes is usually found near the nucleus of an animal cell. It contains a pair of cylindrical structures called centrioles. The cylinders are perpendicular to each other and are surrounded by amorphous substance called pericentriole. Pericentriole material. Each cylinder of centriole is made up of 9 sets of triplets, microtubules made up of tubulin. Evenly spaced triplets are connected to each other by means of non-tubular proteins. Figure 5.18 Structure of centriole. At the proximal end of centriole there is a set of tubules called a hub. The peripheral triplets are connected to hub by means of radial spokes. Due to this, proximal end of the centrioles looks like the cartwheel. The centrosomes helps in assembly of spindle apparatus during cell division. It forms basal body of cilia and flagella. याच्यानंतर एक्झरसाइज सेल स्ट्रक्चर अँड ऑर्गनायझेशनचे. तर आतापर्यंत आपण सेल स्ट्रक्चर अँड ऑर्गनायझेशनमध्ये पाहिलं. सेल पाहिला, त्याच्यानंतर काइंड्स ऑफ सेल्स पाहिलं, त्याच्यानंतर प्रोकॅरियोट्स युकॅरियोटिक सेल्स पाहिलं, कंपोनेंट्स ऑफ युकॅरियोटिक सेल्स, सेल वॉल, मिडल लॅमेला, प्रायमरी वॉल, सेकंडरी वॉल, सेल मेम्ब्रेन, प्लाझ्मा मेम्ब्रेन, सायटोप्लाझ्मा, एंडोप्लाझमिक रेटिक्युलम, गोल्गी कॉम्प्लेक्स, लायझोझोम्स, वॅक्युअस, ब्लॉक प्लास्टिड्स, रायबोझोम्स, हे सर्व आपण पाहिलं. सेल स्ट्रक्चर अँड ऑर्गनायझेशनमध्ये. जर तुम्ही या चॅनलवरती नवीन आला असाल तर प्लीज या चॅनलला लाईक शेअर आणि प्लीज या चॅनलला सबस्क्राईब करून ठेवा आणि बेल लायकन हिट करा, जेणेकरून तुम्हाला आमचे नवीन व्हिडिओज आणि अपडेट्स भेटून जातील. चला तर मग ह्या व्हिडिओचा शेवट इथेच करूया. प्लीज लाईक शेअर अँड सबस्क्राईब. थँक्यू सो मच फॉर वॉचिंग.